Hello, hello. Nice to be back. So the other class tells me they were able to get that record. Where we were staying, we had a really bad signal. Why we didn't have any phone service at all. So I'm glad to see that it got there. So if you, yeah, I basically went out of sequence for a reason because I like to do the protein thing that you're going to see here, kind of up close. I mean, so I can demonstrate it, walk around, do silly stuff, and it's just so terribly important. Everything that I've been saying it, and I can't demonstrate it very, very well, but everything biologically is about shape. We understand the way things work, it's about their shape, as hopefully you will begin to see. It's the hard part for me, and maybe it's the hard part for you. I mean, maybe Dr. Robbins explains it differently when he's in the lab, or the stuff that he gives you. We all, over the years, we've been studying this and being part of it. For me, I've been studying anatomy and learning about anatomy for an excess of 50 years, practicing with it for 25 years, teaching about it for 25 years. You, you think you have a handle on it, trying to have you folks to understand a little bit of it and the relevance is not so easy. And so as I'm trying to do the best I can, and hopefully it gets through, regard to this is terribly important. When it comes to proteins, in short, and I probably said it to you, you are what your proteins say you are. And the next part, when we look at DNA and RNA, explains without going crazy about how we assemble proteins and things like that. That's more of a molecular biology exercise, is to understand that the genetic code is all about making our proteins. What distinguishes us? We're 97 to 98% the same, genetically speaking. We only have maybe a couple of percent difference. And it's those differences is why we appear differently, we have different physical abilities, different intellectual abilities, different capabilities, coordination, whatever. Pick a trait, whatever you want to have. We can do those basic genetic traits, the freckles and the hitchhiker's thumb and all that stuff. Okay? It's all based on this, and it has to do with the shapes of the proteins, or how they're assembled, or how they're not assembled properly, etc. They're a tremendous portion of the cell. I did it out of sequence just to give you an introduction to the cell. And I mean, I'll go back and summarize that when we probably will get through the DNA and RNA nugget. I'd say maybe will certainly be on Wednesday the next time we meet. They have lots of different things that they do. You're going to see a distinction, it's on shape, between structural. We'll probably talk more about enzymes today. Contraction, when it comes to muscles, is something we dedicate. Probably our third unit is almost dedicated entirely to muscles. All those types of things. It's extraordinarily important. What you're going to see is here really the presence of when nitrogen becomes a big player. It's when we begin to see it here. So the nitrogen containing compounds, so far we looked at fats and we looked at, or lipids, and we looked at carbohydrates. Nitrogen didn't really play a role, maybe a little bit when we talked about phospholipids and how they can make lipoproteins. But here, now nitrogen is an integral role, and you'll see it's the same thing when we start looking at RNA and DNA and how they're manufactured. Sulfur is a really important aspect in structural proteins. Structural proteins are different. You learned about them already. I'm going to explain them in a little more detail. Collagen, elastin, reticulin, structural proteins. They make stuff. And they're a little bit different in that regard. They're all assembled the same way. Why one becomes a structural protein, why one becomes what we call a globular protein, which are certainly ones that have the greatest degree of functionality, why one is an antibody, why one is embedded in a membrane. That all has to do with how it's assembled. And it's all a matter of starting with its building block, which is called an amino acid. Unlike glycogen and starch, where everything was glucose. Okay, here the amino acid structure, which I know they have a better, I drew up here, has a central carbon. I'll show you their illustrations better than mine. It has a hydrogen group. Remember, it's got 4 different spots, and they can all tinker toy exchange places. It's got acid on one side, and it has, now you have the amino group, that NH2. The amino group is basic or alkaline. So you have a neutral molecule, even though it's acid on one side and basic on the other side, it makes neutral. Okay, that's all right. What's that R thing? It has 20 different possible R groups. Of those, I forgot to tell the other class that. Remind me, I'll have to tell them. So when you come in here, I beg your pardon on Wednesday. See if I remember to tell them. It's you two. That's your responsibility. If they miss, you're on. You two. Front row. Why would you sit in the front row? You know why I sat in the front row in class? To aggravate the teacher. Stein, are you ever sick? No, that would make your life easy. You all had that kid in school, front row, classes, just giving the teacher miserable time, trying to create humor for humor's sake, commuting to the principal's office. He's the teacher. Goes around, baby comes around. Moving on. So, nevertheless, you have 20 different R groups, 8 of which are essential. What does it mean again to be essential? You have to acquire it through your diet. When we look at enzymes, we're going to talk about minerals and vitamins. They're essential. You have to acquire them through diet. Depending on the author you read, there are 8 or 9. Did I tell you this part yet? You're all looking like this is familiar. There are 8 or 9 we call essential amino acids. 8 or 9 different R groups that we must acquire through diet. You know, I usually put on the test 8 or 9. I won't put them both down, okay, because then that would be unfair because there is a certain amount of disagreement. From those, we can make all the rest of them ourselves. Okay, we can assemble. We have all the building blocks we can assemble. Those we can't. So, and indeed, in food science, anything that when you consume what's called a complete protein, all it means is it has those 8 or 9 essential amino acids. If you ingest, as most people do, animal products or dairy, okay, meat and dairy products, fish, those are by definition complete proteins. So even vegetarians who consume dairy, okay, and frequently do eggs and dairy, whatever your feeling is about that, those are complete proteins. The ones who have problems are vegans. They don't consume any animal products. Okay, so you have to rely on things that have typically a combination of a legume, a bean, which has protein, and a nut or a seed. Hummus, okay, chickpeas, legume, sesame seed paste, tahini. Most famous of all of them, rice and beans. Okay, rice is the seed. Pick a bean. There is one bean. There's probably more than one. There's one famous bean that's a complete protein. Anybody? Asian cuisine. Tofu. Tofu. I'm surrounded by vegans, vegetarians. Drive me crazy. But that's in the family. So I've done a lot of cooking for them. It's fun. Probably do that better than I do the other stuff. Nevertheless, so try to understand that that's the proteins that we ingest help us make all of those. So we're going to look at the bonding, then we're going to look at the structural levels. Voila, here's some of the roles. Notice what happens here. Structural proteins, I can only think of one exception, are typically insoluble. All connective tissue is the single most important one. That's collagen. Notice what makes it strong. It's braided. Okay. Braiding is something we do biologically. This is the shape aspect. Take a shape that's inherently strong. Effectively, what we used to do in surgery, when we would wire bones together, we make a cut in the bone, we reposition it, and I would take what's called monofilament stainless steel, it's very unreactive, suture, and when I was doing it, I'd hold on, we'd fold it, bring the ends together. One had the needle, one had a blank end. The other end, my assistant would put a hemostat on, I'd put a hemostat on my end, we twist them. We braid the monofilament wire, make braided wire. Very easy to do. And then effectively take that, create a hook on the end with the suture itself, drill holes in the bone, and feed it through the way you were sewing something. Just kind of sew it through like little half circles or something. and sew it together. That's how you wire bones together. One of the ways. So that's inherently strong and inherently insoluble. That's important. Enzymes, you're going to see they do a little nugget at the end of this. It's about enzymes. It's about their shape. These areas where, and it doesn't always have to be two things. It could be just one thing that will separate. It could be two things that are coming together. It could be six things that are coming together. It doesn't matter. Every biological reaction requires enzymes, with extraordinarily rare exceptions. And they would typically work, and we talked about that before, both ways. So here's that one we were talking about with the glucose and fructose making sucrose, okay? And they always have the basically the suffix "ase" on them. That implies that means it's an enzyme, and usually tells you what it's working on. Here's a disaccharide-ase that's more generic. And there's lots of other ones that can be oxidases. Don't necessarily imply oxygen. It has to do with redox reaction, oxidation reduction, moving of electrons around. Don't have to know any of that stuff. But it's about the shape. They call this area where they come together the active site. You're going to see when we start talking about denaturing of proteins, that's one of the things that goes. Here, this is a transport protein. Notice it has these in this sort of brownish or tan color. Those are called polypeptide chains. They probably have maybe 70 or so amino acids chained up in each of them. When you put them together, there's well over 200, 250. The definition of a protein is based on the number of amino acids. It's not more important. It's not special. It's no different than a polypeptide. The only way it differs is length. You have 100 or more amino acids, it's protein. Typically, that's accomplished by multiple polypeptide chains. Insulin has 2. Hemoglobin has 4. And what makes this unique is in the middle of each of these polypeptide chains. You learn about this in blood. It's got a little iron protein. It's got an organic iron compound called heme. That's where it gets its name. Heme. The globin are the polypeptide chains, hence hemoglobin. And oxygen can attach itself and be transported. You learn about that in blood and how it transports. It's a very unique, interesting way about it unloads and loads that are there. This is, we dedicate a whole chapter, a whole unit, a whole section to this, contractile proteins. Proteins that change shape and create motion. It's this guy on the bottom. Actin and myosin are found in all your skeletal muscles. Okay? All of those, and all your cardiac muscles. And what they are able to do, that myosin changes shape. When a trigger occurs that causes it to change shape, this head, as it's called, basically goes from here to here, and it attaches to and pulls this other protein called actin. And basically, the actins are attached to either sides of a unit inside this giant cell called the skeletal muscle cell. You're going to learn all about this. You'll be sick of it. Called the sarcolemma or sarcoplasmic membrane, and it pulls it together. We don't contract muscles. We don't bend them. Okay, they shorten, and depending on the shape of the muscle, which you'll learn about from Dr. Robbins, we'll cause certain muscles to do certain things. They'll shorten, and depending on what they're attached to, they'll pull closer together. Some of them will bulge. Mine don't bulge. See, my wife says either they don't like the jokes or you do them so fast they're busy trying to understand. You're throwing in a stupid joke. I know the latter. She's always right. I've survived 74 years by understanding that principle. Gentlemen, I would pay attention to that. Communication. This is insulin. Okay, here's a protein. That's not insulin. That's a membrane protein. Proteins, as you will see, can embed themselves in membrane because some of the areas that make a protein inevitably are fat soluble. So, and again, membrane, the fats just come together. It's easy to stick something else there that's compatible, even though it's enormous. It has an end that sticks out outside the cell and an end that sticks out inside the cell. The insulin, massive, not a little thing like that, big like that, attaches there, and voila, because there's a change on the inside of the cell, that does a variety of things, one of which is to open a doorway for glucose to enter. Okay, and here, lastly, here you can see what they call a defensive. That's an antibody. When you get a cold, okay, when you get strep throat, your immune system samples what's causing the problem. It creates a specialized end called the active site on this. An active site is a recurrent term. An active site on this end of this sort of Y-shaped molecule we call an antibody. It's not the only way we fight infection. And what it does is it attaches to little proteins that protrude. Those proteins are the reverse of that shape. So your body samples it, finds one, creates an antibody to the reverse of that shape, and puts out untold volumes of antibodies. Amazing amount of antibodies once you begin to fight an infection. By sheer numbers, eventually they're going to collide with the virus. They go all over the body. It's not like, oh, we're just going to go with the virus. It's a strep throat. We're all going to the throat, everybody. No. They go all through your system. And what happens is they collide. Temperature does that. It makes them collide faster, increases average kinetic energy, and voila, it attaches. By attaching it, basically it makes that virus a lot easier for your white blood cells to come along and gobble it up. It's not that complicated, and it's a very complex system. You'll learn about that in immune. It's all about shape. The active site shape. The active site shape here that will adapt to insulin, and then it will change shape both on the outside and on the inside and trigger a chain reaction. It'll move to change shape. Okay, it has a unique shape that it will be able to carry oxygen. It has shapes that are specifically designed when molecules come together to help them separate. And it has a shape for strength. Shape. Here we go. Amino acids, 20 different ones. Covalent bonds called peptide bonds. A very special bond, very strong. And basically, you're going to see it's a joining of the amine side and the acid side. Okay, and that's what makes it sort of a unique molecule in that regard. That's better than my horrible drawing. That's what varies. So if we put two of them together, okay, same slide there, I actually have sequence. Here you can see what happens. They could all be the same. They could all be different. There is an unlimited number of polypeptides and proteins because there's no rules. You could have 100 that are all the same, 50 and 50, 49 and 51, and they could all be different. You can use 2 of one, 63 of the other, 5 of this. You get the idea. Unlimited number of possibilities that are there. I mean, I don't know what that, because effectively they have an unlimited shape, so I would assume it continues to be unlimited. The R group's influence. So for argument's sake, we'll say they're the same, doesn't matter. What happens is that water splits off from the OH of the acid side and the H of the amine side. The water splits off and we form a bond between carbon and nitrogen. That is the peptide bond. So it's COO bonded to nitrogen with an H. You don't have to know the chemistry, just that it's peptide bonds. And it's reversible with the proper enzymes. When we ingest proteins, for whatever reason, whatever we eat, our digestive tract can only absorb the individual amino acids. So eventually, when they get to the lining of your small intestine, they have enzymes in the exterior of the individual cells. Fancy name, they're called brush border enzymes. You'll learn about that in digestive. They break them down to the individual amino acids and we bring them in. That's how it works. So here are the levels of structure. It's all based on primary. Primary is the linear sequence of amino acids, the batting order. Voila. Batting order. It forms a secondary based on, this is important stuff, hydrogen bonds. As you will see, here's the primary structure at a minimum, without even considering what the R groups are, and they could be all sorts of different. There are really a myriad of possibilities with R groups. They can be acidic, basic, positively charged, negatively charged. All of those, if that's what they are, would be water soluble. And there's ones that are just fat soluble. One of the R groups is nothing more than another hydrogen. See, and you have to know all the, you were doing biochemistry class in that room after us. Then, the pre-med and bio majors have to learn all of those things. They have to know all 20 and what their different properties are, because that's super important. You guys just have to be familiar with the basics. And so what will happen is it will reliably go into two folds. I only do this one. It will either form a pleated sheet. Understand, this is so important. When your protein's folding, or what allows your brain to fit in your skull, that 30 feet of digestive tract to fit into this little area from your diaphragm down. It's all got to be folded. Everything. You're going to see that your stomach, basically, normally unfilled is about a couple of ounces. It can stretch out because it's filled with folds. It's a fancy name you'll learn about in anatomy in AP2 called erugai, which just means ridges. And the ridges are fold. Your bladder has the same thing. When it's stretched, they stretch out. When it's empty, the folds become apparent. So it's just like accordion pleats. It's all about the shape. It's the old joke. My wife says, Lou, get into shape. And I look at her and go, round, it's a shape. And then she throws stuff at me. I smile out here in front. It's an old joke. Don't even start. Nobody appreciates it. Hydrogen bonds. They either make it here and hold it together, or they form what are called alpha helices. That's a beta pleated sheet. And so these are right-handed twists. It's left-handed for beta sheets. But we don't have to go into that. And so all of those together are held by hydrogen bonds. That's the secondary structure. If we move on, the tertiary structure, the next level, involves combinations of these. The vast majority of these, if you looked at them all in number, would form what we call globular proteins. This one is forming a particular structure we call a globule, and let's say that might be an active site. The way that generally they're a little more complex than that, where something could attach. So it's a combination of pleated sheets and helices that come together. As you will see when you get to fibrous, it's all helices. And then in order to most of the globular proteins are like this. So you took two of these that are very, very similar. You don't have to know the name. It's called transthyretin. It's a protein that attaches to a very small molecule called thyroid hormone. Because thyroid hormone is fat soluble, and it needs to have a protein that it can bond with to carry it in your bloodstream. Your bloodstream is obviously water soluble. And thyroid hormone goes to all of your cells. What's interesting is, it's proteins because they have this kind of construction and those kind of properties. You're going to have ones that are going to have aspects that are going to be water soluble and dissolve in the plasma, ones that are be fat soluble. A lot of different globulins is a general name. You're going to look at these, they're called beta globulins, particularly transport things or transport proteins in your bloodstream. And this is one that transports thyroid hormone. So you have all of those different levels that are there. So tertiary, why do they form a globule in a particular shape? It's all here. So for the secondary structure of hydrogen bonds, the R groups are based on attraction or repulsion. Acids and bases come together to neutralize each other. Positive and negatives attract to neutralize. Water soluble areas come together. Fat soluble areas come together. When you're talking about a protein that's got 100 amino acids or more, or multiple polypeptide chains, you're going to have all of those factors. That's why they can sit inside of a membrane and have protruding areas that are both fat soluble, or internal areas that are fat soluble, and exterior areas that are water soluble. It's about the shape. Okay? And so quaternary means 2 or more polypeptides. Again, hemoglobin has 4, for example, insulin has 2. Insulin is inactive when it's secreted or not active. And then when we chop it up a little bit and it becomes active, when it works in your body to metabolize sugars that are there. You don't nothing out of this. What happens to those globular proteins? Okay. Globular proteins, just like anything else, and I'll skip where the fibers is, compact, spherical, water soluble. These are all throughout your bloodstream, sensitive to environmental changes. Read that mostly as temperature and pH. They're designed to function around a pH of around 7.4, 7.35 to 7.45, and a temperature around 37 degrees Celsius, normal. Variations in that area cause them to be less efficient. A little bit of temperature, like I said, increases collisions molecularly, and therefore it will cause reactions to speed up. So that's all it is. It's a series. It's not like magically something looks for each other. Chemicals don't have any eyes. They don't have any attraction. It's all about numbers. How do viruses and bacteria hurt us? Numbers. How do we deal with them? Numbers. Now, antibiotics are nice, but you take 6 to 10 days for you to ramp up making antibodies at first. That's why you take antibiotics for 7 to 10 days. You'll learn about that in my. So specific functional regions are called active sites. And what happens when you have sufficient variations, mostly increased acidity, lowering the pH, or increasing temperature, it starts to lose its shape. We have a fancy word, we call it denaturing. Its shape is its nature, okay? What it's able to do. So whether it's an antibody, whether it's a hormone like insulin, whether it's a chaperone, and basically just with the name, it escorts other molecules to the appropriate place. Think of what happens when we assemble amino acids to come together to make polypeptides and proteins. We're not going to go into that. That's molecular biology, all that type of stuff. It's interesting. Depending on who teaches you micro, they may actually teach you that. I do not. I think that's more for biology. No one will ask you if you're a health science major or a nursing student, about AP and E sites and how they interact on the platform of the ribosome. I try. It sounds like I'm making it complicated. I'm trying to make it easy. So all of those things and enzymes, basically all about their shape. So when you denature them, effectively you take this mass, and if it's too much heat or variations in pH, or we're starting to freeze them, that mass, that globule, that form. So think of what it can do. At normal temperature, I told you what insulin does, it takes that membrane protein. The membrane protein makes a chain reaction. One of the things it does is it changes shape, the insulin. It causes a door that's closed, molecularly speaking, to open. So glucose can come through. It alters the shape of a transport protein in the membrane. You're going to learn about it. Basically, some transport proteins are always open. It might close the door. In the case of insulin, it opens the door for glucose. When the insulin detaches, the door shuts. All about shape. It's there. So why do these irregularities happen? It begins to lose its nature. And eventually it's going to lose its globular shape. Eventually it's going to lose its individual shapes. It's not going to come together. All those positives and negatives, acids and bases, water and fat soluble. And it's going to start to lose its secondary structure. But what will always remain, everybody, what's left, primary structure. Those peptide bonds are extraordinarily strong. It takes specific enzymes and very specific conditions in your small intestine, first your stomach and then your small intestine, to break them. So what's always left when you denature, okay, it unfolds, changes its three-dimensional shape. We'll get to the fibers, proteins. The active sites become deactivated. Most frequently, increased acidity. It could be the other way around. That's rare. Increased temperature. And typically those, what they're describing there is most likely irreversible changes. Okay. Freezing of all of those. When something gets too cold, okay, it just stops moving around as much. And it can't change that shape. But we can retain its function if it unfreezes. And historically, anecdotally, meaning everybody in medicine, you hear about people who fall through a frozen pond. We can resuscitate. Their proteins don't irreversibly denature if you can get to them, let's say, It used to be 40 minutes. I don't know if that's changed with the technology of today. You would always try to resuscitate. You would not resuscitate. Use that double negative. That's there because there's just too many people who have survived and not been damaged by it, remarkably enough. So the whole idea, what makes you reverse event, is the extremes. So pH is here, around that 7.4. Okay, so this would be increasing, going alkaline to basic, acid, and it's very much a bell curve. So the optimal way is around 7.4, our average pH in our bloodstream. That's what we call the optimal function. We have a pH optimum. It starts to go down, it's like a slippery slope. Very hard to go in this direction. You can't see a lot of alkalosis, juicy, diabetes. One of the bad things when they become acidotic, when the sugar's out of control, and that means proteins stop doing their job, enzymes. Temperature's worse. See, there you have some degree of reversibility. Temperature is not a bell curve. It starts that way from cold, but when it gets to your optimal temperature, 37, it's like falling off a cliff. I mean, you go to 42, not a whole lot different. You're gonna die. 108 degrees Fahrenheit. That's all, and that's really important. It's why in science, when we sterilize things, what do we use? Temperature. Vast majority of things we sterilize, it's called an autoclave, microlab, and micro, you'll learn all about that stuff. And so irreversible. I'm not on cooking an egg. I'd like my steak a little more rare. Sorry, pay for this one. We'll cook another. I do the same routine all the time. And it changes shape. Everything is about the shape. That's there. And so and I'll get back to that in a second. Let me back up. Which brings us to the globular ones in general are water soluble. There are probably a few examples that are not. The structural ones are designed to be insoluble. The only one and surely the best example, maybe there's another one that I don't recall off the top of my head, is fibrinogen. Don't have to know it yet. That's the one in your blood. It's in a precursor form. You'll learn that's what ogen means. And what happened, the whole thing with your clotting cascade, whenever you cut yourself, whenever you scrape a knee, you activate the clotting cascade. The whole point of it is to change that fibrinogen from soluble, dissolved in your bloodstream, to insoluble. Effectively, it forms a fishnet, molecularly speaking. There's a wonderful illustration in this textbook about that. In other textbooks, you can find it. It's like a fishnet. It traps red blood cells and platelets and firms up, and that's how your blood clots. All the other ones, the collagen you learned about with connective tissues, the elastin, the reticulin, effectively, they are basically a series of those spirals, okay? Strand-like, mostly tertiary, some quaternary. And basically, it's like this. Why do we braid things? If you have a spiral like this in and of itself, okay, it's strong, but even I can do that. But if you braid it together, I'm not faking it. One of you guys could tear it, not me. You get the idea? I certainly go to zero expense to show you these demonstrations. We have another one coming when we start looking at phagocytosis. And I use something that I always carry in my back pocket. You saw a picture of the dog. I don't leave home with that. 74-pound Irish Doodle available to you cheap. Just take my dog, please. We were away. We had issues. The cat was indignant. We left the door open because he likes to go outside and inside. We got home for 12 hours. We didn't see him. We thought he crosses the street. He sees a naughty kitty. Okay. We figured, oh my God, something terrible has happened. He begs the neighbors for food. He's one of those. We left the door open. We watched it. I get a note this morning. My wife was up till midnight. I'd be up before 6. She goes, his name's Cloud, the white one. Cloud, I went upstairs to go to bed at midnight. And there he was on the bed. How he got in, he was hiding the whole time. He was mad at us. We weren't there to service his needs. It's a cat, basically. Absolutely true. So that's strand-like, water-insoluble, tertiary, some quaternary if they're more complex, mechanical support, all the ones you learned about, collagen, number one on the hit parade. The contractile ones can change shape. They're a little bit more complicated. We'll get to myosin, you will see that. Which leaves us, and probably the last big topic, are enzymes and enzyme activity. Enzymes are catalysts. They are globular proteins. And what they do is about their shape. Well, a little bit before. It has something, you can usually draw it like this. It has an active site. As a site, it could be designed for one thing separated or multiple things to put together in any combination thereof. It has something that fits in there. That's got a fancy name. You'll see both of these, sometimes called the ligand, sometimes it's called the substrate. It could be very small, it could be fairly large. The protein inevitably is very, very large. What happens when it starts to denature? Effectively, that active site begins to change its shape. So I'll take away the ligand, even that could be another protein like insulin. And all of a sudden we'll get, now that rounded end can't fit, square peg, right? It alters its shape. So immediately you can begin to see the shape dictates the nature of the problem. Every biological reaction, I mean, I can't even think of one off the top of my head. When you're talking about our body putting things together and taking it apart, requires an enzyme. They are called catalysts. And the terminology we use, we say it catalyzes the reaction, to put it in verbal form. And all they do, yes, they regulate it by their presence or absence. And a lot of times that's what certain pathways produce. But it's all about making it faster. And the way it does it, this is a test question. It lowers activation energy. Every biological, it's not magic that they come together or come apart. You have to put some energy into the system. We call that the energy of the activation. How do we determine if it releases energy or stores energy? If it breaks bonds to release energy or puts together bonds to store energy, we are able to assess that by we sort of add up how much energy we have to put in, how much energy did we get out. Enzymes don't change that. If it's going to be releasing energy by breaking bonds, we're going to release energy. Or so it just makes it happen faster. It's just like when we increase temperature, we get more collisions. This is vital because, as they'll tell you here, you can get millions of reactions in a minute. Thousands upon thousands in seconds of these things can occur. Just an immense amount, and the enzymes don't get consumed. So they lower the activation energy, as you can see. This is what it looks like. We'll get there. So effectively, this is the energy of activation without, and all it does is it makes it easier. I mean, we all use the ski slope type of thing. I mean, when imagine climbing up to the top of a hill that you're going to ski down on. I mean, think of the energy that's necessary. Think of that as potential energy. When you ski down, the energy is being released as you lower it. How do we, not being so exhausted, we can't have a ski lift. Something that enables it. That's what the enzymes do. They reduce the requirement to effectively get to the top of the hill and allow the reaction to proceed. And then we determine what was the overall energy to start, what's the energy that ends up, whether or not we're going to, again, store energy as making bonds or release energy. That's breaking them. So that's how it works there. So most enzymes we call holo or whole enzymes. They have the protein portion, fancy name apoenzyme. I'm not worried about holoenzymes and apoenzymes, but I do want you to know about cofactors, which are metal or metal-containing compounds. We have a name for them, minerals. They too are essential. You have to consume them through diet. Even a teeny tiny amount is sufficient. Vitamins are coenzymes. So what typically happens to assemble that protein, go back to the way it looked. But to assemble that protein properly, to make that opening, okay, it's going to make that active site whole. Somewhere over here might require a mineral. Here, because it's organic, that would be a vitamin. Mineral will probably just have to attach to it in some way, shape. Here, most likely, that vitamin helps to complete or make the active site more functional. It doesn't really matter. Just as you all know that cofactors are inorganic and minerals, and coenzymes are vitamins and they're organic. Okay, coenzymes, organic, vitamins, for the most part. I mean, they're all in the vitamin category that are there. When you hear about folic acid, well, it's not a vitamin. It's not vitamin A or B or C. It's in the category of B vitamins. So they're very, very specific. Insulin is only going to bond to an insulin receptor. All cells have them, pretty much. It's a very highly specific substrate. So it's got to have that receptor in order to take it. We use the term ASE to indicate what they catalyze, hydrolases involving water, oxidases. Typically in our world, we see them with oxygen, but it really applies to redox reactions, oxidation, losing electrons. Just so happens oxygen is where we part that we lose. And so here's the effect. They call it priming the reaction. Sounds reasonable. Substrate binds. It forms and it's very we used to use the lock and key model, but we figured out years ago that that's really not the case. Lock and key meant, well, it's going to tightly bind to that active site, and then when it's ready, it'll release it. Really what happens is there's subtle changes. There are changes that occur in the active site, and as the ligand or substrate gets close to it, it begins to cause molecular changes in both of those areas. That's it. So it's got a fancy name that you probably don't have to know for me, called induced fit. Like when something gets closer to that active site, it begins to induce a change in that active site. So as it goes farther away, it'll go back to normal. So the idea, rather than a lock and a key, it may be a key getting stuck in the lock. It's very quickly. You can just change, grab it, boom, let it go. Grab it, let it go. That's the way I would characterize it. It's there. Oh, we saw that. And they're just showing you. Here, the one we saw before. Here, they're creating. Side note. Do not let this confuse you. Here we have two amino acids with an enzyme making a complex, forming a peptide bond. Notice they call this a dipeptide. This is one of those things in biological science in biochemistry that bothers us. If I say dipeptide, di is for two, you expect there to be two peptide bonds. There's not. It's really two amino acids and one peptide bond. This falls under the category of the just because rule. There's no logic to it. Tripeptides, that's right, have three amino acids and two peptide bonds. Thankfully, after that, they're all polypeptides. Once they get to 100 of them, either as more than one chain or multiple chains, we have a protein. This is annoying. I didn't make it up. It is annoying. And really, that's the part of this that's so important. The enzymes are everywhere. You can, what would be a classic genetic disease, since we have a minute or two left? One of the earliest genetic disease testing that was done was for a disease that is called Tay-Sachs disease. It's called autosomal recessive. I mentioned that to you before. Of people of what's called Ashkenazi Jewish birth. My first wife and I came from Eastern European Jewish ancestors. There's a fair number of those how to carry that recessive trait. And basically what happens if you both carry the trait, there's no way to have to be what we call dominant, what we call homozygous dominant, because you wouldn't survive past age 5. You both carry the trait, there's a 1 in 4 chance. Typical genetics. So we were tested, neither one of us had it. It was one of the thoughts we were having when we were trying to conceive children biologically. There was a lot of genetic basis. Now there's multiple genetic tests that are available. This was one we had done at McGee in 1980, 45 years ago. Tay-Sachs accounts for an enzyme that causes the assembly and breakdown of a protein fatty complex in your brain. Without that enzyme, breakdown products are stored in the wrong area in your nervous system in the brain, and your brain begins to lose function. The baby develops normally for the first year, smiling, happy, doing all those landmarks that you had, crawling around, maybe starting to walk, and all of a sudden it begins to deteriorate. By anywhere between age 2 and age 5, it stops. It's extraordinarily difficult, and that is one of the criteria when people were deciding whether or not to have children. And whatever or carry a child to term, that's a whole other issue, not for discussion here. But understand that's something like that, an isolated gene like that accounts for a protein lipid or protein fat complex that affects the myelin sheath in the brain. And the fact that we can't get rid of a particular waste product is enough to cause a normal baby, to every parent just explode with that baby to unfortunately succumb sometime between age 2 and age 5. I will leave you with that. We're going to do the nucleic acids, DNA and RNA days are upon us. Good day. Have fun.